So this week we're going to do some instrumentation, both the stuff that we use already in the micro lab and then stuff that's sort of, well, it's already invented, but it's coming down the pike, and whether or not it'll be used depends really on the budget and the needs of the laboratory. So what's the reason for instrumentation, in particular in the microbiology lab? Since, a lot of the tests that we've been doing them for manually for so long. Well, there are several things. Obviously, speed is a big thing. We want to be able to get the most accurate results to the physician as quickly as possible to ensure that the patient outcome is as, and that it's positively affected. And obviously, speed would be a feature for that. The limiting factor in microbiology, of course, is the fact that the bugs have to grow, right? We can't make them grow faster. However, maybe we don't need to look at their growth directly. Maybe there are molecular ways that we can look at genes that are produced, proteins that are present, things like that, and get an ID in that by that method instead. So speed is something that's definitely still on the table for microbiology. It's always good that we can improve speed. Reproducibility is always going to be the thing that really we are looking at when we talk about instrumentation. There's a lot of things that we can do manually. They're just as fast. An instrument's not necessarily going to make them faster. But reproducibility is something that's important. So for example, if you're going to do, just think of the little tests that you do on your desk. A catalase doesn't take any time at all, a couple of seconds for you to do a catalase. However, your method of doing a catalase might be slightly different from somebody else's method of doing a catalase. And that's something that CLSI really doesn't, they're trying to reduce that. We want to make sure that everybody does it exactly the same way. Use the same amount of organism, the same amount of catalase, allow the incubation to occur at the same rate, never pick up any of the agar, because remember the agar will interfere, all of those things. If we can make that reproducible so that the test is performed exactly the same way every single time, we can be more assured that the result will be the same. Okay, so obviously reproducibility is important to us, and it's usually one of the main drivers behind instrumentation. Data retention is another thing. Data mining is a huge, getting all, managing all the data that you have is important. It's not just important for the patient records, it's also important for billing. It's also important for epidemiology, infection control. It's important for ordering of tests and reagents so that we know, well, how much reagent do we use specifically. All of that data is actually very important, and it can frequently lead to a reduction in cost, which is, of course, one of the things. Every hospital is trying to reduce costs at this point. Instrumentation doesn't always reduce costs, so you do have to, you do have to sort of weigh the differences. Am I getting so much more benefit on the speed, reproducibility, data retention side that it actually justifies the cost that you're going to include in this. Now, no matter what you do, when you're talking about bringing in a new machine, validation is key. You must undergo validation in order for you to bring in any machine. And so what validation is you compare the instrument, the new, whatever it is, the new procedure, the new instrument, against two things. You compare it against the manufacturer's claims, and you also compare it, well, actually three things we'll say. Manufacturer's claims, expected results, right? So industry-wide expected results. And also, if you had a previous procedure, for example, if your instrumentation, you have to compare it to your manual results as well. And you have to see some kind of correlation between those instruments, between the instrument, the new procedure, and your old procedure or manual method. You're not allowed to just, hey, I got a new machine, I'm going to plug it in, let's start using it. And in order to do that, remember all that crazy math that we had to do. You have to figure out the accuracy, you have to figure out the precision, you have to figure out the analytic sensitivity, the specificity, the reportable range, all of those things, and also a reference range. Now, those are things that you would probably more likely apply to, say, a chemistry machine, right? Reportable ranges and stuff like that. In micro, we're really looking at sensitivity, specificity, and then, of course, reproducibility and correlation with our manual methods. There's no real reportable range, right? In micro, you're not really supposed to have pathogens growing in you. So when we find one, obviously, the reportable range is you're not supposed to be there. And a reference range, you know, again, if we can prove an ID by a new method, the reference range, you would actually compare libraries. So the library of the manual method might be, well, catalase positive equals staph. If we determine a different way to instrument, create an instrument that'll, you know, with a new test that'll say, this is how I'm going to determine staph species versus another species, you have to validate them. The catalase works how many times to tell me that this is definitely staph, and this new method works how many times, you know, what percentage of the time is it accurate in telling me that this is staph. So that's the kind of math that you have to do. It's required, and you have to keep that validation on record for the life of your hospital and for the life of that machine. Now, you know, you've been through the, you know, or you're going through the micro rotation, you know that there are already machines there that we use, and they have made our lives in the micro lab much better, okay? And that would have to start with the blood culture instrumentation, and not just blood culture. Now they have mycobacteria and fungus as well. I can tell you because I'm old enough to have done manual blood cultures, but the use of an automated blood culture system to detect bacteria growing inside the liquid has really improved the life of the microbiologist. Basically, we have a couple of major systems out there, BacTech and BactElert, and I'm not you know, endorsing either one, and there are others I'm sure out there. These are the two most familiar, most used in the micro lab. Generally speaking, the way these work is the machine can sense things like a change in pH or a change in the headspace pressure in the instrument in the bottle, right? So they might be able to do that. Those are common things that they can use. BacTech actually uses at the base of the bottle, there's a sensor that has a dye in it. And that dye reacts with CO2 in the bottle. Okay, so what happens is the amount, that sensor, the dye there, will modulate the amount of light that'll be absorbed by a fluorescent material in the sensor. So you have kind of, the fluorescent material acts as kind of a substrate in the bottle. The CO2 reacts with the dye, which then interferes with the ability of that fluorescent material to absorb light. And there's a photodetector at the base of each station. So, where's my pen? So at the back of each one of these holes, and actually sometimes you can see it, a light will flash, and that light is flashing into that fluorescent material. And then as the bacteria grows, the CO2 is released by the organism, which of course that changes. That'll interact with the dye in the base of the bottle, which will change the ability of that fluorescent material to absorb light. And what you have over time, still has to be measured over time. The BacTech, the BactElert, both of them take measurements approximately every 10 minutes. So over time, it'll take readings. And as the CO2 is released and the fluorescence is interfered with, you'll get a graph, and the computer predicts the point at which that graph, the change of or the interference means that there might be growth in there, and it'll flag that bottle as positive. What that means is that if you're looking at this entire unit down here, my pen back, you know, maybe just a few of them will flag positive. Maybe these ones will flag positive. And then you pull those, you take a gram stain, you see if there's any bacteria in them. What that means is you don't have to pull the rest of them. You let the machine follow the rest of them. Okay, so it really reduces the amount of work that we have to do. Yes, in the manual days, and yes, I had to live through it. We used to have to, gram stain all of them, or we would actually do a blind subculture. All of them. You know, you do every single aerobic bottle, you'd have to manually view every single bottle so that you could see if something was growing in there. So it just reduces your tech time incredibly. And that, in reducing tech time, major money savings, okay? Those are the things that, that's how BACTEC works. BacTalert works just a slightly different method in that instead of having a fluorescent sensor in the base of the bottle, they actually have a colorimetric method where as the gases are produced by the bacteria, they'll bind to these indicators in the base of the bottle, and that'll generate a color change that actually you can see. The machine will read it, but you can actually see it too. And actually, it's not uncommon for, well, at least in my experience, we used to look at the bottom of the bottle when we would get it down from the floor. And, you know, sometimes it takes, you know, half an hour or so, you know, even up to an hour or two to get those bottles from the floor. And if you have a gram-negative rod in there, it'll already be colored. You'll already see the color change. So don't even put it in the machine. Take it out and gram stain it. Okay, but it does allow for very quick reactions. And one of the reasons is the colorimetric method, instead of just relying on CO2 and O2, it actually relies on several volatile gases and organic compounds that are released by bacteria, gram negatives being a big number of them. And so colorimetric method and BacTalert is slightly more sensitive to a wider range of gases that are produced, or volatile compounds, I should say, that are produced. And so it gives you pretty quick results. Both of these are lovely. They both work great. And the key thing is that they both reduce your tech time massively so that you don't have a tech who's literally just assigned to subbing bottles all day long, okay? Also what it does is it increases the speed at which we detect those positives. For me to visually see a change in the turbidity of a blood culture bottle, that might take literally a full 24 to 48 hours, whereas BacTec and BacTalert can probably detect those changes at a much quicker rate. So they might detect a change in 10 to 36 hours, much faster than the manual methods, okay? And then some of the things that both systems provide are these specialized or the bottle types that support the growth of the various bugs. So you already know that every bug culture should include two types of bottles, an aerobic bottle and an anaerobic bottle. And that allows the conditions for that particular organism to be best replicated. But then you also have to have other types of bottles, and there are several other types of bottles. You have pediatric bottles, of course, because where the anaerobic and the aerobic bottles, those we expect to get between 8 and 10 mils in each bottle, okay? But in a baby, you can't really collect between 8 and 10 mils. So for a low volume draw, like in a baby, but also some of your geriatric patients might require these, but definitely in a baby, you would do a pediatric bottle. If you're doing from a line, you can always get between 8 and 10, so you should always do the double bottles. But from a peripheral, blood, that's really difficult to get. You might need to do a pediatric bottle. We also have antibiotic neutralization. They sell special bottles, or they sell the additive, one or the other. I forget which one. I think BacteriAlert has a special bottle. Bactec sells the additive. It's just a charcoal additive. It's nothing very fancy, but it'll neutralize the antibiotics if the patient is already on antibiotics. And that's important because insurance doesn't pay if you collect the blood culture bottles after antibiotics have been started. So by having that charcoal neutralization, you can argue with the insurance company that, hey, we took steps to make sure that the antibiotics were not part of this culture. So that's important to remember, and it's something that all labs really should consider keeping on as part of their stock, and let the doctors know that it's there. And then also you need a whole nother bottle. Although most fungus can grow in your routine anaerobic and aerobic bottles, if you're specifically looking for fungus, and remember, fungus you keep for a very long time. So the longer you keep a blood culture bottle in there, the more likely it is to grow something, which would be skin flora associated with phlebotomy. If you really do want to keep a fungus bottle for 4 weeks, you should use a specific fungus blood culture bottle so that it can inhibit the growth of normal bacteria. And then, of course, we have mycobacterial bacterium bottles. That's usually a whole different machine. BacteriAlert and Bactec both make one. You have the midget system, and you have the BacTB or something like that. I forget the name of it. It's a fancy name. But basically, those are machines and bottles that are specifically tailored to the very long incubation times for Mycobacterium. And of course, it includes antibiotics to keep down normal flora and things like that. The specimens still have to be, it's not like you can just collect a sputum specimen and inject it into a Mycobacterium bottle. You actually have to still concentrate the specimen, acidify the specimen, all that stuff. You have to do the normal concentration procedure, and then you inject it into the bottle, put the bottle in the machine. But it is usually at this point used to enhance the manual methods, because the manual methods you don't get growth for 7, 14 days, whereas in the instrument we do get growth literally 48 to 96 hours. We've seen growth from a sputum, so that's pretty good recovery, much faster. And when you're talking about TB, you really want it fast, so that you get the patient into precautions, but also isolate the patient is what I'm trying to say. Precautions. There are difficulties with each system. There are requirements that we have now by CLSI and JCO. For example, we are supposed to verify the volume of each bottle. So you have to verify that there's actually between 8 and 10 mils in each bottle. That's hard to do if you're not collecting it. BacteriAlert actually is introducing a weight recording function in their machines, so that that's how you're going to verify the volume. So obviously every distinct volume has a distinct weight. So by verifying the weight of the bottle, you can ensure that whether it's a low draw or not. And also we are required to maintain a record of the phlebotomists who drew these, which would then we would associate with contamination. So you use those phlebotomy initials that are supposed to be on every bottle, and then you're supposed to record them, and you're supposed to correlate those initials with the with any contamination that might occur. In some hospitals, the LAS system will take care of that, so the phlebotomist registers the fact that they drew that blood culture bottle, and then if contamination occurs on that, you can correlate it. We used to have to do it manually, no kidding. We used to have to write down the actual phlebotomist initials. Sometimes you can't read those initials. That's what we did. But again, BacteriAlert has an idea for this. They are actually going to take a digital picture of every single label so that you have the date and time recorded that's supposed to be written on every label, as well as the phlebotomy initials. So that's pretty interesting technology that's added to, some people might think it's superfluous, but as somebody who had to write down those stupid initials and look at every stupid bottle, yeah, I think that's a great idea. And these help us to meet some of the JCO requirements that go with bottle handling. Actually, also I'll go with specimen collection procedures, contamination rates, and all of and training people who aren't apparently cleaning the site properly. So those are things that allow us in the lab to meet those JCO standards across the entire hospital. So this might affect nurses. You know, if you find that, you know, Nurse Ratchet up on the ICU keeps contaminating the bottles, well, Nurse Ratchet needs retraining. Some new things, some innovations are moving forward, some of the products that are sort of coming down the pike. You guys are already familiar with the MaldiToff. One of the other areas that is very interesting is adding microarrays to the bottom of those bottles. So MaldiToff is a whole new system, and it doesn't use bottles, but with microarrays, we can use the same bottles that we're all used to. But instead of just having a colorimetric system that responds to any volatile acid, they actually changed the colorimetric system into a microarray so that you specifically know, if you look in this cartoon up here, you specifically know which, you know, look at all the different things that we have up here, the different volatile acids and indicators that we can have in this microarray, and this is just one example. Then as the organism grows, what we're measuring, if you look down here in this cartoon here, what we're measuring is the rate of production of the various volatile acids in the number of hours. So you're combining both the type of volatile, volatile gases produced or whatever indicators or dyes are produced, as well as the rate at which they're produced. Now you can compare those against a new library that allows you not only to know that that the bottle is positive, but what the most likely growing organism is in there. So that's pretty good information. So you're still growing up the organism, which we always need to do in microbiology because we're going to have to do a sensitivity. We want that organism on a plate. But we're also getting an ID right away from that blood culture bottle. This is going to be faster than the MaldiToff because the MaldiToff requires that you grow it on a plate first. This is giving us an ID before it's ever on a plate. That's pretty cool. Okay, so they're working on innovating this. They got a lot of work to do with the libraries to make sure that the libraries are validated because these would be new and different libraries compared to the ones that we use in IDR. Ours are based on growth over, 18 to 24 hours, even up to 46 hours. So these libraries are reading, look at this, in 6.3 hours. You've got 18 hours. These are reading pretty quickly. So that's, the kind of stuff that we're looking for. And you can see the comparison that they're making between this is just one bottle here. So these are the gases produced at 7 hours. Or I'm sorry, it's read in this direction. So at 6.3 hours, we have these gases. At 7.7 hours, look at this, we have the next gases, okay? So, and so on down the line. Very interesting stuff coming down the pike. How cool would it be to know, okay, this bottle's positive, but I already know that it's just a diphtheroid. I'm not going to freak out about it. I'll let the doctor know right away, okay, it's positive, but it's a diphtheroid. You might not really have to worry about it. Obviously, we still have our routine identification plates. The microscan identification system is probably one of the most widely used automated identification systems, and it's very simple. It uses a microtiter plate method, very simple. It just takes all of the reactions that we do, the big tube reactions, all of those reactions, shrinks them down into a microtiter plate. No big thing, okay? You've got all your sugar fermentations, and you can see those here in this box are the sugar fermentations, and then we have our enzymes and our drug reactions. Here's all our enzymes in these boxes here, okay, or most of our enzymes anyway, some of them. No, most of them are enzymes. And then our drug interactions. We've got the rest of the plate are drug interactions. And those antibiotics and susceptibilities are used to help expand the libraries and get more confidence in the identification that we use. There are some great things about Microscan. First of all, it's very easy to set up, right? I'm sure that you've seen this many times, pardon me, where you create a suspension of your organism in these little bottles. You pour it into a dispensing plate. You use your Renoc system to draw up the exact amount of this suspension and inoculate all of the tubes at the same time. So here's your Renoc right here. She's, I think that's a woman, is drawing up the same amount of specimen in the little pipettes that will correlate with each of these wells up here. And she'll just bring that over and she'll inoculate this plate. And then the plates are put in. You can put these these plates into an automated or a manual read system, which means that in some places we incubate the plates overnight in an incubator. We manually add the reagents the next day, and we use what's called an autoscan system, where we can, one plate at a time, we read the actual microscan plate. We develop a biocode, and that's compared against the library by the computer. And the computer will read the color changes. Or we can use a walkaway system where the instrument adds all of the reactions at the appropriate time. So it'll keep the time on the plate. How long has it been incubated? It'll add the reagents at the appropriate time and read them at the appropriate time from them. Obviously, the walkaway is easier on the tech, reduces a whole lot of tech time, and it also allows you to read a large number of these plates very quickly without bothering the tech. The autoscan it's there, it works fine, but it's a little bit more clunky, obviously, because you got to add everything yourself and all that stuff. Okay, so, but the microscan system, it works very well. Then we have the Vitex system, which is, it's actually quite similar to the microscan. Basically, it's shrunk down all of your tests even more. This is a microscan card, I'm sorry, Vitex card here, and each of these little wells contains a different reagent or potentially a sugar or an antibiotic, or, well, this is an ID card, so there could be one or two antibiotics. This will contain all the reagents needed for that particular identification panel, the enzymes, the sugars, all that fun stuff, okay? And these are basically intended, you have, if you look at this little cartoon over here, this tube has the suspension of your bacteria, whatever your a suspected organism is. And you have a small tube going up from the suspension. It's a vacuum tube, basically, into the card. You place this hole, and you can put as many cards on this carrier here as you need, okay? And then you place this entire carrier into a vacuum reader, and the suspension is drawn up through the tube and through all of these wells in order, okay? So that you end up filling every single one of these wells inside the vacuum chamber all the way. So the order in which these reagents and sugars are placed in that card has been figured out so that one doesn't react with the other. Okay, so but all of them are filled, and you can kind of see they don't go in exactly this way. You can see the little tubes down here and how you see these little lines down here and how it's drawn up. Those are the things, the actual pathways that the that the solution follows. So then once your card is filled, you put it in the instrument, and this, similar to the microscan, the instrument will read, it'll measure the time that that card was added to the instrument, and at the appropriate incubation points, it will read the reactions, and it can read the reactions at several different incubation points, so that if one test should have been read at 10 hours and the next at 14 hours and so on, it can keep track of that. Automated read for the Vitek system is the most common. However, it is possible to manually read these cards, and you can input your read or adjust the read into the computer. That's just less common. Some of the cards, when they first came out, were all manually, like their yeast ID system when it first came out was only manual read. Their anaerobe identification was only manual read, but they've adjusted that now so that they can be read in an automated fashion. So they're basically the same. One's a little bit smaller than the other. One of the benefits, I would say, of the Vitek system is that you don't have to add no reagents are added. So the reagents are all built into those little tiny bubbles, those little pockets for the solution. Now, as far as the antibiotics and the susceptibilities that we read off of the same systems, Microscan, you can get a sensitivity. Vitek, it would be a different card, but you get a sensitivity as well. We need to talk just for a moment, and I know we've talked about this before, between the difference between breakpoint and true MIC. Okay, and I know you've heard of this before. And I'm going to start with true MIC because I think you need to understand what that is first before you understand what a breakpoint is. So the, true MIC requires that a serial dilution of your antibiotic be created. So you create a serial dilution, and then you add a known suspension of your specimen. So in this case here, where we have a tube dilution made, this can be in a microscan well, this can be in a Vitek card, doesn't matter, or it can be in real tubes. Sometimes we are called on to do this manually for special testing in micro. But basically, you start out with the dilutions of your drug, in this case ampicillin, and then you add the same amount of specimen of your specimen suspension to each one of these wells, and you look for growth or lack of growth over the next day. Obviously, if it grew in that dilution, that's not enough. So you ignore everything that becomes cloudy, okay? And then you look at the ones that aren't cloudy, where no growth occurred, and you can read the level at which, let's say this is the first one that didn't grow, then your MIC is 32, and the drug is resistant to the bug. I'm sorry, is resistant to ampicillin. Okay. This is much more accurate. The true MIC is considered the gold standard. We want a true MIC as often as possible. However, as you can tell, it's obviously going to be more expensive because you have to do the more tubes. More tubes means more expensive. So the breakpoint was developed, and the breakpoint is certainly a perfectly legitimate way to do things. It uses a single or a limited dilution, and the dilutions that are used are based on the levels that are achievable in the patient blood. So, if I'm doing a tube dilution and I have, say, a level of 2, and I find out that my specimen is sensitive all the way to an MIC of 2, well, that's great. But you can't achieve a level of 2 in the blood, okay? Same thing on the other side, resistant all the way to 128. Okay, that's great, but you can't achieve that level in the blood. So really, are these numbers useful? Well, for those proponents of the breakpoint, which definitely makes sense, 8 and 16 are the levels that are achievable in the patient at the site. So let's see if that works. Let's just use let's just use those levels, those dilutions, and see if the bug is sensitive or resistant. You still use the same interpretations. So if it doesn't grow at 8, so no growth at 8, let me erase some of this stuff here. No growth at 8, you would record this as sensitive. If it grows at 8, however, then 16 becomes your interpretation, and you say, well, this is intermediate. And if it grows in both of them, you'd say it's resistant. That's how you play this game. Okay, now obviously this is a little bit less sensitive only in the fact that certain, especially the emerging conditions like CRES and ESBLs and things like that, those frequently you need a full tube dilution. You need to have a full MIC in order to properly interpret whether or not you have an emerging pathogen here with antibiotic resistance developing. However, there's nothing wrong with reporting a breakpoint as long as it is actually reported as a breakpoint. You cannot misrepresent it as a true MIC because it's not. Okay, you didn't see growth in any of the other wells. You don't know exactly what the true MIC is. So it should be reported as a breakpoint. That is something that many hospitals do have a problem with, many systems have a problem with, because even on the microscan, for example, some of the drugs are true tube dilutions, they're full MICs, and some of them are breakpoints. But most hospitals just report everything as an MIC, and that's not truly accurate. So that definitely is something that should be addressed in order to be, fully transparent with our doctors. Now, some of the other really fancy things that are coming out are automated plating systems, and I think you need to know what these look like. The key thing being, look here, the difference. This is our standard quad plate right here, and this is an automated a specimen that's been plated by a machine. Okay, and you can see they don't look the same. What's the difference between the two? Well, automated systems work very well for liquid specimens, not so much for swab. We haven't figured out how to do the swab one in an automated means right now, but it works very well for liquid specimens. And as you know, the number one specimen coming into most micro labs is, what is it? It's urine, okay, which is a liquid specimen. So that can easily be taken care of. If we don't have to plate any of our urines, we can let a machine do it, and we're assured that they're all plated exactly the same. Oh, that's something that's important. Okay, so with automated plating, we know that what the machine does is it takes the exact same amount. The exact same amount of the specimen, and it plates it at a point in the on the plate, right? So if you set that you want exactly, half a mil of specimen on that plate, then that's what it'll put there. You'll set the parameters. Your lab will set the procedures and the parameters for that instrument, and then it uses it uses this comb here looking thing. You see this here, a new one, obviously sterile for each one, and it drags it along in a circle around the plate, which allows for the dispersal of all the organisms. And you can see it doesn't look the same as our quad plate, but we do still get similar results. The quadrants might not be as apparent, but you can see where it starts. And so from the start, you just make your quadrants in the normal, mathematical way. Just split the plate into quadrants. And you can still see we have very nicely isolated colonies out in the fourth quadrant for us to work with. Okay, and so this allows these labels, and this is just a picture of what one looks like. And from the top, you can see it's pretty busy. There's a lot of stuff going on here. But when you add, barcode readers and label makers and things like that, look at, you're just loading your urine cups onto this, queue, and the machine takes care of the rest of it. And these are cups. If you have the actual tubes, the BD tubes, it makes it even easier, right? The machine will sterilely enter the tube, so you have a much more confidence in the cleanliness of your plating. Okay, and again, what's the number one specimen coming into every microlab? Urines. That's the number one. So this would take a lot of tech time away from you. It's a very expensive instrument, but a lot of tech time would be managed. And there are hospitals that are using it. This is not just some fancy thing. There are hospitals, large volume hospitals, that are using it. And then we have also coming up the pike, automated plate reading. Now this makes someone like me who grew up manually, and I really like to see the plates, a little nervous, but I can see the benefits of it, okay? Basically, what you have here is something that the microlab never had. Okay, we never had evidence of what we actually saw. So if I read a culture today and I look back in the patient's history and I say, gee, oh, Susie Q read a culture from the same site 3 days ago. She saw no growth, but look at me, I'm seeing all this strep pneumo. I wonder what she really saw, if it was really, the same specimen. We don't have, we never had that kind of luxury in micro, because once you read the plates, they were gone. So especially if there were no growth. So with automated plate reading, what you have is the computer scans every single plate in that culture at the same time. So if it's a sputum, it'll look at the blood, the MAC, and the chocolate all at once. If it's a urine, whatever your plates are for that particular culture type. And it'll keep an image of that. It'll keep a digital record, right? A digital picture of what grew on that. So if you can look I keep going to the wrong place. If you look up here, you can see the plates for that particular incubation, right? Scan 1, what grew? Scan 2, what grew? What does that mean? Okay, day 1, here's what grew. Day 2, here's what grew. You set all of these parameters. How long am I going to keep these? If these are, well, these look more like throats or urines or something like that. I can't really see it chocolate. This looks like a urine plate. I'm not sure. But anyway, it'll keep a record of this, and it'll keep that record with the patient file. So if you look here, down here, you can see, oh, I'm looking at all the plates for that patient, for all the specimens that came down on that patient, and I can compare. Okay, she had the lactose fermenter here, still has it here, and you go on. You can compare the growth. Is it getting better? Is it getting worse? However, you want to compare it over time. For the most part, what's going to happen with the digital reading, the automated plate reading, is all of your negatives will be eliminated, right? So you don't have to spend time with those negative plates. No growth, get rid of it. No growth, get rid of it, or re-incubate it. If it didn't grow, then the instrument will, and these automated plate readers are hooked up to your incubators. In some cases, the plates never leave the incubator. Okay, they're kept in the incubator the entire time. Okay, so the instrument reads the plates and says, okay, we got to keep this one for another 24 hours. Keep taking pictures. Okay, and for other ones, it'll say, wait, I'm going to flag this as positive and release it to the tech, and the tech can work up the specimen. Okay, this is a very different kind of workflow pattern than that we're used to, and it allows the microbiology lab to become patient-centered instead of specimen-centered. And what I mean by that is you look at one specimen at a time on any one patient. So if you have a sputum specimen in front of you, you're looking at that sputum specimen. And it would take initiative on the part of the technologist to look into the history if the patient has historical sputum cultures, whether they are close in the same visit or previous visits. Okay, when you're talking about automated plate reading, you get to look at all the plates of that specimen type on that patient at the same time, and so it allows you to have better reference for that patient. You know, if let's stick with our sputum specimen. If that sputum is on a CF patient and it's the first one of this visit, but the last visit of that patient, which was 3 months ago, that patient had a serious problem with Pseudomonas aeruginosa, and you see it here on this plate, you can bring it to the doctor's attention. Oh, it's back. That Pseudomonas is here. It doesn't belong here. Okay, and so it allows you to change the workflow from that, one specimen at a time kind of thinking to one patient at a time, which hopefully will improve patient outcomes. And at the same time, of course, having all these digital images allows us to collaborate over time, over space and time with other hospitals, with pathologists, with other professionals. It gets us second opinions or assistance from our colleagues. Do you think I should work this up? And one of the things, of course, they're really playing is, look, they can send it to the doctor's iPhone or iPad. Hey, what do you think? You think this is good? Of course, I would want it to go to an Android, but that's just me. Okay. And it also allows us to incorporate e-learning. There's a database already, the Hugh database of culture plates, so that the database is there. We can compare, and if you look in this little picture here, we're comparing the radiographic films with the plates of the specimen collected at the same time that the films were taken. This just allows a much more collegial atmosphere across the hospital and across the industry when it comes to microbiology. It's not just, well, my hospital does it this way and your hospital does it that way. It's like, hey, let's all look at this. We're trying to treat this patient the best we can. Is it an expensive endeavor? Yes, it is. But the fact that you wouldn't have to look at any of those no-growths, I see that as a great cost savings in tech time. And then, of course, we have to move to molecular testing because there's a lot more molecular testing in the microbiology lab than there ever was before. Most of it directed against emerging infections. If you don't have screening protocols in place for these emerging infections, meaning your MRSAs, your VREs, ESBLs, CRESs, all of those, then you're not serving your patients well. There's several ways you can screen, but the molecular methods are things that hospitals really favor because we really appreciate the sensitivity and the specificity of those. We know that MRSA screening, we have PCR techniques that can identify the genes that allow us to identify MRSA in patients before they're ever placed in a room so that we know whether or not they need to be on contact precautions. Same thing with VRE screening. VRE screening, there's less molecular because it's not as big a problem in most hospitals. It tends to be localized. So that one probably is less, there are molecular tests out there, but it's probably less likely molecular. And then certainly ESBL, we're looking for CRES screening as well to ensure that we get patients on contact precautions as soon as possible. Hospitals are also moving to private rooms for everybody so that contact precautions just becomes normal for everybody, so that the cleaning that you expect somebody to do when they go into a precautions room actually will probably apply to every single room on the floor at some point, especially on certain floors, when you're talking about ICU and really serious floors where patients are at high risk. And then we also have molecular testing for pathogen identification. We already do it for C. diff. Again, that's a precautions issue. We want to be able to identify strep pneumo, especially in the elderly, as soon as possible because they are so highly susceptible to pneumonia, and unfortunately death from strep pneumonia is also a problem. Staph aureus, because we want to be able to identify it, is the #1 pathogen that's isolated in the laboratory. Wound infections, toxic shock syndrome, scalding, so all those things are ideal. We also, you may think, why do we care about Staph epididymis? Well, if we can identify an organism as epididymis really quickly, then the doctor might be able to say, oh, well, it's just contamination. I don't need to worry about it. And a lot of that pathogen identification, in particular from blood cultures, is used is very highly useful in with MALDI-TOF, as you know, and we'll get into that. And then also there's PCR fingerprinting, where we do actual gene sequencing of bacteria that's very highly useful for epidemiological purposes. Okay, so if you need to trace an outbreak in your hospital, if you need to identify the actual strain, specific strain of a particular, maybe your patient comes in with, I don't know, a VRE. I gotta go up here. Maybe your patient comes in with a VRE and you want to actually identify the specific fingerprint, right, molecular fingerprint of that strain, to ensure that that doesn't go, so that if another patient shows up with VRE in your hospital, you can trace it back. Is this an outbreak? Where can I trace this to? Is it occurring through, some of our staff? Do they need to be retrained? Any of that stuff, okay? The molecular testing is definitely more sensitive, and it allows us to get more information quicker. And you can see this little cartoon here. We have all the different identification methods. You have everything from your API, very manual, to your MALDI-TOF, okay? Highly sensitive to your phenotypic array. And then we still need to do more stuff. Okay, I know what it is, but I need to know its sensitivity. Is it a MRSA, VRE, CRES? What's its sequence if I need to track it? All of these things are necessary in some cases.